this is part 3 of the trilogy of nitric oxide pathway as a new drug target for Duchenne muscular dystrophy. So let's talk about clinical trials now. And we were talking last about oral sodium nitrate beetroot juice. And the question is, will better blood flow preserve muscle? So we're going to, thanks to pilot funding from Coalition Duchenne here in Los Angeles, we are going to, instead of having one dose of the beetroot juice and measuring blood flow after a single sport shot 3 hours later, we're going to give, we have patients that are going to take the beetroot juice 3 times a day, one shot 3 times a day for 6 months to see if we can improve their maximal grip strength, to try to translate the 9-month preclinical trial from Raju's lab in Washington, D.C. To actual, from mice to patients. And we want to show that we can have the good effect of improving maximal grip strength and bulking the muscle without adverse side effects from too much of nitric oxide. The worry is that it could be too much of a good thing. We don't know the dose. Too much nitric oxide might produce hypotension, and so the safety monitoring is with 24-hour ambulatory blood pressure monitoring, and also measuring blood pressure when the subject's patients lying down, sitting up, and standing up. Again, I don't think this is going to be a big issue because we believe that the nitrate is delivering nitric oxide in the microvessels for oxygen exchange, not in the resistance vessels that regulate blood pressure. And then the key worry is, are we going to worsen nitrosoactive stress in the skeletal muscle? Dong Shenduan and others are very concerned that cytosolic NOS, which is not being regulated appropriately and continuously releasing NO in the wrong place, could nitrosylate bad targets, such as the ryanodine receptor, that could exacerbate the calcium handling abnormality and could also increase nitrosoactive stress and inflammation. We're going to do 2 muscle biopsies, baseline and after 3 months, and we're doing proteomics in those biopsies to study nitrosylation of a whole variety and phosphorylation of a whole variety of targets. And it's a collaboration with Rochelle's lab at UCLA and with Jenny Van Eyck's lab at Cedars-Sinai, runs our proteomics core. We're using this very sensitive hand grip dynamometer, which is designed specifically for weak patients to study maximum grip strength before and after the daily treatment with beetroot juice. So our 2-hit hypothesis, to recapitulate, is that the first hit in dystrophin-deficient muscle is a lack of this structural support, and that is exacerbated, if you would add insult to injury, by functional muscle ischemia. So there's unregulated excessive alpha-adrenergic vasoconstriction causing ischemic insult to these vulnerable muscle tissue with repeated bouts of use-dependent injury that would accelerate the destruction of muscle. That's the 2-hit hypothesis. And the hope is that with either an NO donor or PD5 inhibitor, we could slow disease progression. So going back to Yvonne Kobayashi's famous paper in Nature, here she used a fancy MRI technique. It's T2 imaging. I'm not an imager, but what this does is it suppresses the fat signal to amplify the water signal. Just a cross-section of the gastrocnemius, well, the hind limb muscle in the MDX mouse. And most of the tissue is gray at rest. Immediately post-exercise, you see these arrows showing white areas of white spots, and that is labile water thought to be inappropriate muscle edema. Pretreatment with Dalafil rescues the phenotype and prevents this excessive free water signal in the muscle, suggesting that there's less injury to the muscle tissue and less water leaking out of the muscle cells into the interstitium. And so we're trying, and so here, if you look in the wild-type mouse, or even in the nNOS null mouse, there's almost no post-exercise T2 signal by MRI. And the MDX mouse says that 30% of the muscle volume is now white or with labile water, and tadalafil almost completely rescues that, one dose before this downhill bout of exercise. So now can we translate that to patients? And this is unpublished work in progress. So here is a patient. If you look at the bottom of the slide first, this shows that this is a patient with common deletion of exons 45 through 47. There's no nNOS binding site, and there's no nNOS around the sarcolemma. In the untreated patient, this is now a higher level of hand grip exercise. It's about 50% of maximum, 1 second on, 1 second off. This is the cross-section now of the forearm. And what you see is that most of the muscle is gray by 3T MRI. After 2 hours later, you start to see these abnormal accumulations of water, very much like Yvonne saw in the hind limb of the MDX mouse. Now, it's preserved. That signal is still there 24 hours later, and in 48 hours it's starting to go away, but not completely. So Krista Vanderborn, who's our colleague and an expert on this T2 MRI in Gainesville, Florida, said, after a marathon, if someone develops edema in their muscles, it's going to take days to go away. So if you believe this is really injury, then that's what you should see, and this is certainly consistent with that. Fortunately, a week later, when we brought the patient back for the tadalafil experiment, the abnormal water signal had completely resolved. We pre-treated this patient with 20 milligrams of tadalafil, and now we greatly attenuated that, if you will, post-exercise muscle edema. So this is N of 1, degrees of freedom equals 0. If we can replicate this multiple times, that'll be extremely exciting. And then here, by comparison, this is another Becker patient. This is one of the cousins that has this splice mutation, exon 46, and has preserved sarcolemmal nNOS, really a normal amount, and he's got normal grip strength. And when he exercised, there's no water signal accumulating. So it really does start to support Yvonne's work from the MDX mouse and translate it to human patients, which is what my lab is all about. So on this cartoon, this is what we think is going on. So again, we think that in Becker, that there's a short dystrophin. In Duchenne, there's no dystrophin. nNOS is misplaced to the cytosol, where not only is it absence of a good thing, but it may be a bad thing to cause nitrosoactive stress. But the little nitric oxide, the residual stuff that's dribbling out of the muscle cell into the vascular smooth muscle area, that the downstream target, cyclic G, can be boosted by tadalafil, and that rescues sympatholysis, prevents post-exercise vasospasm. Hopefully it would cause better endurance and block the injury to the muscle by muscle-specific troponin, which Jenny Van Eyck's lab actually is the first to develop that assay. So that's what we're working on next. But this is all intermediate phenotyping except for the endurance. So now it brings us to randomized, double-blind, placebo-controlled phase 3 registration trial of tadalafil to repurpose this for Duchenne muscular dystrophy. This is an international trial sponsored by Eli Lilly, that's the manufacturer of tadalafil. So I'm honored to be the PI and the chair of the steering committee in this trial. And this is the culmination now for me of 3 decades of research to get to this point. So the inclusion criteria for the trial, these are boys with Duchenne. It's not mutation specific, which is very different from exon skipping, which only about, roughly 10 to 12% of the boys, depending on the mutation, would be eligible. so cuts a wide swath. This age is 7 to 14. The boys have to be ambulatory because the primary outcome is a 6-minute walk distance. The left ventricular ejection fraction, a crude, standard clinical measure of left ventricular cardiac systolic function, has to be normal, and they're on have to be on stable steroid therapy because the glucocorticoids have such a powerful effect on prolonging ambulation. It's a standard of care now, so we want to add this on top of the standard of care. Can we improve the current standards? So this is the boys are randomized to in double-blind fashion to either placebo or a low or high dose of tadalafil. Remember, we used 0.5 and 1.0 milligrams per kilogram in our blood flow studies. Well, because tadalafil has such a long half-life, it accumulates in plasma, and the folks at Lilly have so much experience with this that their best guess was that slightly lower doses than 0.5 and 1 would basically be equivalent to that of a single dose. And so this goes on for 48 weeks, and then there's an open label phase. So the initial goal was to enroll 306 boys in 62 study sites around the world. I'm happy to say that we exceeded our enrollment target, and we enrolled 331 boys. And the trial, the last patient will complete the 48-week primary outcome in the first quarter of 2016. So my colleagues and I will be analyzing the data spring of next year, so fingers and toes crossed. The hypothesis is that once daily oral tadalafil for 48 weeks will lessen the decline in ambulatory ability, which is measured by the 6-minute walk distance compared to placebo. Again, I mentioned there are 62 study sites, and that we have more than 306 boys around the world enrolled. I wish we had something better than the 6-minute walk distance because there's no doubt that there's a volitional component to it. But at the moment, this is sort of the gold standard for regulatory approval by the Food and Drug Administration. And just as an aside, my good friend Craig McDonnell at UC Davis has really been the pioneer in doing the best we can to quantify the 6-minute walk distance and pushing the envelope to develop new and better regulatory outcomes for neuromuscular disease. On the study endpoints, the 6-minute walk distance I mentioned is a primary, but we have lots of secondary outcomes that are, again, time function tests, the North Star ambulatory assessment, standard test, as well as quality of life, which is key in this condition. Very excited about is this exploratory outcome, performance of upper limb scale. So this is upper limb quantitative assessment of activities of daily living for muscle strength and function. That would be great because this could be transitioned to non-ambulatory patients after the age of 15, for which right now there are almost no clinical trials, although that's changing. Pulmonary function test also, as you know, the muscle that's affected the most in this condition is the diaphragm. So very excited about the sub-studies. My lab has the only, at Cedars, has the only blood flow sub-study where we're doing our sympatholysis protocol and post-exercise hyperemia with Doppler. Krista Vandenborn has a wonderful collaborative MRI imaging consortium that's funded by an R01 mechanism from NIAMS, the branch of one of the two main branches of the NIH that funds muscular dystrophy research. So it's been a triumvirate of U Florida, Gainesville, University of Pennsylvania, Children's Hospital, Philly, Oregon Health Science University, and they were generous enough to come to Cedars and sync our imaging sequences so that we could be the fourth member of this, what had been a triumvirate on this particular study. So those are the sites doing the leg MRI. It's a T2 MRI protocol, very much like the T2 MRI imaging I showed you in the pilot studies on Becker. Then we also have a cardiac MRI sub-study where we should have, I don't know the exact number, somewhere between 50 and 80 patients. So I'm hoping that we'll have enough sample size. This will be the first trial ever to use the T2 leg MRI signal as a quantitative measure, not just of natural history progression, but of the interventional study. And this is only the second study to use cardiac MRI as a treatment endpoint. So in closing, to summarize these three lectures, what about the NO pathway as a novel treatment target? Well, we're repurposing existing drugs, so this can happen a lot faster and a lot cheaper than starting from scratch, bringing a drug to market. You know, the drug companies always get a bad rap at every political season, but the flip side of that is that it takes about $1 billion to bring a drug to market, and the vast majority of them fail. So the drugs that act on the NO pathway, they're not going to cure Duchenne or Becker. The hope is they would slow disease progression. What are the best drugs to restore NO signaling? I mentioned that the PD-5 inhibitors happen to be convenient and have a good safety profile. We'll see if the NO donors are better. There are also new now G cyclase inhibitors that are coming to market, so time will tell. The key question is, will the therapy slow disease progression? My friends in pediatric neurology and in muscle biology say, all right, Ron, you've showed umpteen times and convinced us that sarcomal nNOS is important in vascular regulation, because if you don't have it, vascular regulation is impaired, and you can bring it back with PD-5 inhibitors or NO donors. Does that have anything to do with natural history of the disease? And if you recreate the normal blood flow phenotype, will you slow disease progression? Well, we'll have the answer after the end of this massive phase 3 tadalafil trial sponsored by Eli Lilly. It'll be very informative. If respiratory and cardiac muscles are also benefited, that'll be a home run, because these are the major causes of disability and major causes of death in this disease. This could be an adjunct to gene therapy. You know, the exon skipping that's being actively pursued here in the Center for Duchenne Muscular Dystrophy at UCLA. The purpose is really to convert the Duchenne phenotype into a Becker one, right? To start producing some dystrophin. And interestingly, the Becker patients foreshadow what we might expect from exon skipping therapy. And depending on what exon is being skipped, the exon skipping may or may not restore proper NO signaling. So this could very well be combination therapy, that if the exon skip does not restore the RS16, 17 binding site, that then using a PD-5 inhibitor NO donor may be great combination therapy. And then another final member of the Kevin Campbell diaspora, Ronnie Cohn, did some beautiful studies showing that sarcomal nNOS is either that nNOS is either completely lost in the sarcolemma or mainly lost in the sarcolemma in a variety of neuromuscular diseases, even with there's an appropriate dystrophin background. So even bed rest or physical inactivity in a rat or a mouse for a period of time, for a week or so, causes nNOS to dissociate from the sarcolemma. So it could be that, you know, even after someone breaks a bone and is casted for a while, that you can imagine that restoring NO modulation might hasten recovery of the muscle. So for a whole lot of neuromuscular diseases, this work could possibly be generalized beyond Duchenne and Becker. So the initial rat study that in terms of our funding, the initial rat study was funded by the National Institutes of Health, the National Heart Lung Blood Institute. It was part of Dr. Jerry Mitchell's 40-year long program project grant on exercise and cardiovascular regulation. And then that was at UT Southwestern. As I said, my ideas about this lay dormant for a while. And then moving to Cedars and collaborating at UCLA, we did the first proof of concept studies thanks to funding from the Muscular Dystrophy Association Parent Project Muscular starting in 2010. And that teed up a clinical trial planning grant by NIAMS in 2012 in collaboration with Lee Sweeney and our colleagues at University of Florida, Gainesville in Oregon, as well as Craig McDonald at UC Davis. So we started to tee up a phase 2B trial, but it was so expensive to do even a phase 2B, and fortunately Eli Lilly stepped up to the plate. So NIH teed up the trial, and then Eli Lilly took it over for this very large phase 3 randomized controlled trial, for which we will have the outcome data in the first or second quarter of 2016. So once we got into it, things moved very rapidly into phase 3 trials. Thank you so much for your attention, and best of luck in your careers.